All right, let's dive right in. Today, we're exploring the incredible, microscopic world of sperm. Understanding the science behind how we analyze and prepare sperm is absolutely fundamental to the success of modern fertility treatments, everything from IUI to IVF. So, let's break down that whole clinical journey from start to finish.
Okay, let's start with a number that might surprise you. In at least half, that's 50% of couples who are looking for fertility treatment, there's a male factor that's contributing to the issue. So this is not some niche problem. It's a huge piece of the fertility puzzle, which is why a really solid understanding is so important for everyone involved.
So here's the journey we're going to take in this explainer. First, we'll cover the absolute basics of semen analysis. Then we'll take a closer look at what makes a quality sperm, its form and its function. After that, we'll get into the lab and see how sperm is prepped for assisted reproductive technologies, or ART. And we'll wrap up by talking about a condition called azoospermia, what happens when there's no sperm found in the ejaculate at all.
Okay, Section 1. This is the foundation. It's ground zero for the entire male fertility workup. Seriously, without an accurate, standardized semen analysis, every single step that follows could be built on shaky ground. This is where it all begins.
There's a reason this process is so incredibly precise. The World Health Organization has laid out these really strict guidelines because every little detail matters. That 3 to 5 day abstinence period, that's to make sure the sample is truly representative. Producing it on site, that prevents temperature shock, which can literally kill the sperm. And that last step, incubating it at body temp, is crucial for the sample to liquefy so you can even analyze it properly.
So what exactly are labs looking for? Well, these are the WHO's lower reference limits. Now, this is super important. These are not pass-fail scores. Think of it this way. They represent the 5th percentile of a fertile population. So if you took 100 men who had recently fathered a child, 95 of them would have numbers at or above these levels. It's a baseline, a reference point, not a hard line for who is and isn't fertile.
All right, so we've got the basic numbers, the count. But honestly, that's just scratching the surface. Now we have to dig deeper and look at the quality of the sperm themselves. It's not just about how many there are, it's about how well they're built and how well they can actually do their job.
So take a look at this. This chart shows us how sperm motility, or movement, is graded. What clinicians really care about is what's called progressive motility. Are the sperm actually moving forward, making progress? They get graded from A, the Michael Phelps of the group, swimming fast and straight, all the way down to D, which are completely immotile. That forward motion is everything. Without it, a sperm has zero chance of reaching an egg on its own.
Next up is morphology, which is just the sperm's shape. And here's a fact that might blow your mind. Even in a sample from a very fertile man, the huge majority of sperm are actually abnormally shaped. It's totally normal. This slide shows you some of the common defects: heads that are too big or too small, messed up midpieces, or bent tails. The real challenge is finding that tiny percentage of sperm that are shaped perfectly.
So we've looked at the outside of the sperm, right? The count, the movement, the shape. But what about the most critical component, the thing we can't see with a regular microscope? What about the quality of the precious genetic cargo it's carrying?
This is where we get into something called DNA fragmentation. The DNA inside a sperm head is packed incredibly tight, but sometimes that packaging process goes wrong, or the DNA can get damaged by things like oxidative stress, which can come from lifestyle factors like smoking or exposure to toxins. And here's the bottom line. When that DNA gets too damaged, it dramatically lowers the chances of a successful pregnancy.
Okay, so we've got our analysis. The next stop is the lab, where the sample gets prepared for treatment. The whole point here is to find and isolate the A-team, the small group of superstar sperm that are the most motile, functional, and ready for action in either IUI or IVF.
And here's why that's so crucial. Raw ejaculate just isn't suitable for assisted conception. The seminal plasma it's in actually contains stuff that can stop fertilization from happening, not to mention all the debris and dead cells. So, sperm preparation, which is sometimes called sperm washing, is all about cleaning up the sample and concentrating the best of the best.
And there are two main ways to do this. You've got the swim-up technique, which is pretty elegant and works great for high-quality samples, and then you have density gradient centrifugation. Think of this one as a more active filtering process, which makes it the go-to method for more challenging samples that might have low motility or a lot of junk in them.
So let's walk through the swim-up. You just gently place a clean culture medium right on top of the semen sample, and then you wait. It's that simple. The healthiest, strongest sperm will literally swim out of the gunk and up into that clean layer all on their own. You then just collect that top layer, and boom, you have a self-selected group of the most vigorous sperm.
Now, the density gradient is a bit more complex, but it's also incredibly powerful. The best way to think of it is like an obstacle course or a filter. You create these layers of special media in a tube, and when you spin it down in a centrifuge, only the healthy, strong sperm have the power to push through all those dense layers. They form a clean little pellet at the bottom, while everything else, the dead sperm, the debris, gets trapped in the layers above.
All right, for our final section, we're going to tackle one of the most challenging situations in male infertility, azoospermia. This is what it's called when, even after a thorough analysis, there is simply no sperm found in the ejaculate.
So the definition seems pretty straightforward, right? But the diagnosis is actually a very rigorous process. A sample only gets the official azoospermatic label after it's been spun in a centrifuge at high speed to concentrate anything that might be in there. And even then, after looking at the resulting pellet under a microscope, they still find 0 sperm.
Now, azoospermia isn't just one thing. It has different root causes. You can think of it in 3 ways. Pretesticular means there's a problem with the hormonal signals from the brain to the testes. Testicular means the factory itself, the testes, isn't making sperm correctly. And post-testicular, or obstructive, means the factory is working fine, but there's a roadblock preventing the sperm from getting out. Figuring out which one it is absolutely key.
For men with this condition, there's still hope for biological children thanks to surgical retrieval. With PISA, a very fine needle is used to pull sperm directly from the epididymis, which is where they mature. This works really well for those obstructive cases. TC, on the other hand, is a biopsy where a tiny piece of the testis itself is removed. This can be a total game changer for non-obstructive cases, because even if the factory has mostly shut down, there might still be little pockets of sperm production they can find and use for IVF.
And that brings us to our final thought for the day. The science has come so far. We've gone from using ejaculated sperm to sperm from the epididymis to sperm from the testes, and now even to using precursor cells. As our technology gets better and better, allowing us to achieve fertilization with cells that are less and less mature, it really pushes us into some complex new ethical territory. It's a question that science, medicine, and all of us as a society will have to keep asking as this amazing field continues to advance.